Hey guys, in this unit we're going to be looking at some fairly in-depth and more complex STEM fields that some of you may or may not be familiar with, depending on your background. So I wanted to take a few minutes and just kind of go through some of the scientific terms, the jargon if you will, that you're going to be seeing both in your reading and in some of the discussions that I've posted for you to watch. And I just wanted to take a moment to define these and keep them clear so that you can follow along with that material as closely as possible. So I'm going to start by defining genetically modified organism, which we're going to see a lot. And you're going to see this most commonly referred to as just a GMO, whether we're talking about crops or animals. Now, a genetically modified organism is broadly just referred or used to refer to any organism that's produced through one of several different genetic engineering techniques. And commonly is used to reference organisms that have DNA or genetic material that is not naturally found within that species. So that brings us to our next two terms. These are two terms that are used to kind of clarify this discussion about GMOs a little bit. And this is the term cisgenesis and transgenesis, or cisgenetic and transgenetic. Now, the root cis just means same or similar, and trans means different. Okay, so a cisgenetic crop or cisgenesis as a whole is used to refer to the production of a GMO containing DNA from two closely related species. While transgenesis is used to reference a GMO containing DNA from two unrelated species. Now, you'll also hear transgenesis used as a common term, just used to broadly refer to the creation of any GMO. But when you see it in context with this cisgenesis term, it's used to specify the difference. That is, whether we're talking about DNA from closely related species or distantly or unrelated species. Now, the process also matters a little bit here. So cisgenesis is also used to refer to a species that could be produced by traditional selective breeding methods. So it could be produced traditionally. Whereas transgenesis is used to refer to a GMO that could not be produced using traditional methods. All right, I'm just going to take a second to clean up the board here, and then we're going to move through our next set of terms, which is just a couple of different ways in which GMOs can be produced, or new products can be produced. All right, guys, the next two terms we're going to look at are something that I want to just take a brief moment to discuss so that we can understand the differences between them more clearly. So we're going to talk a little bit about mutational breeding, as opposed to genetic engineering. So mutational breeding is at this point, kind of classical technique, started after World War II, that uses mutagens to introduce mutations within a seed bank or seed pool from which you can then select to get a beneficial trait out when you're looking for a new organism or a new crop. So we're just going to say this uses mutagens. And mutagens can be either chemical mutagens or radiation. And so if you take a seed and you expose it to some mutagen, so let's say we shine in some X-ray radiation, and we do this to a bunch of seeds, we're going to be introducing a whole bunch of random genetic mutations to that population, and then we can grow some of those seeds and assess the progeny for the presence of traits that we're interested in. And if we find, say, some drought tolerance here, we can then take that plant and we can either grow it, or we can mutate it further, or we can outcross it to another current crop to try and isolate that trait. But in this way, we have isolated a new organism, or a new crop, if you will, and these guys may either die or have non-beneficial mutations. Now, this technique is generally defended as not producing GMOs. And the reason for that is that when this technique is used or introduced, it is introducing mutations to the existing DNA of that organism. It's not introducing new DNA from an outside source. Now, genetic engineering is creation of a GMO, creating a genetically modified organism through one of several techniques. And those techniques are used to introduce external DNA to your target organism. So we're just going to say through several techniques. And these techniques can include things like vector integration, a gene gun, or targeted endonucleases, such as zinc finger, Talon, or CRISPR-Cas. Which we're going to get into in just a second. So if you will give me a minute to clean off this board, I'll be back with you in a moment and we'll go through our last few terms for this discussion. All right, guys, so the last set of terms that I wanted to kind of go through for a little bit of clarification are some of the targeted restriction endonucleases that you're going to see in your reading. So I'm going to start by just defining what an endonuclease is. An endonuclease is an enzyme that cuts DNA, but it cuts it specifically anywhere other than the end. So think of it cutting in the middle. I'll just say in the middle, but it's really anywhere that's not at the end of a DNA molecule. And for the most part, the endonucleases that we're going to see if we have a double-stranded sequence of DNA are introducing double-stranded breaks. So they're going to clip all the way through in order to introduce a break in DNA that's going to be responsible for the repair machinery introducing the transgene of interest. Now, there are kind of three categories of these types of targeted restriction endonucleases that we're going to see, or that you're going to see referred to. These are zinc finger endonucleases, Talon systems, and CRISPR systems, specifically CRISPR-Cas. Oops. So zinc finger restriction endonucleases are a type of endonuclease that are constructs that involve the fusing of a zinc finger binding domain. So if we have our double-stranded DNA, we would have a zinc finger recognition domain that is going to recognize a specific sequence of DNA, and it's going to be fused to an endonuclease domain, which is going to cut that DNA adjacent to that targeted binding sequence. Now, TALEN stands for transcription activator-like effector nuclease. So we have our TALEN, and the transcription activator-like, or TAL domains, are the DNA binding domains in this construct. So this would be our TAL domain that is combined or fused to an effector nuclease domain, which is responsible for introducing the break in the DNA strand. Now, CRISPR stands for clustered, regularly interspaced short palindromic repeats. And it is the CRISPR domain, or the CRISPR system, that is responsible for binding DNA. Now, CRISPR is a family of DNA sequences that are in prokaryotes that are derived from viruses and are responsible for providing an adaptive or a form of adaptive immunity for prokaryotes in recognizing foreign genetic material. Now, this is the CRISPR domain here, and these are typically paired with the effective nuclease domain. The most common one is the Cas9 restriction endonuclease, which is why you'll hear this commonly referred to as CRISPR-Cas or CRISPR-Cas9. All right, guys, hopefully that helps to clarify a little bit of the jargon or the terminology that you're going to see over the next couple of weeks. I hope this helps to clarify a little bit of these more complicated terms. And if you have any questions, please shoot me an email or give me a call.